So, good morning, everyone, and thank you for joining us to our webinar, What Lies Beneath, a comprehensive look at extractables and leachables, presented by Neotron. We start with the first speaker presentation. First, I present myself. I'm Valeria Mazzoni. I'm your host for today's session. I'm the scientific project coordinator for Extractable Leachable project. I have an industrial biotechnology degree, and I've been working in Neutron since 2017. Next, I have the pleasure to present you Mattia Albano, the QC team leader for contaminants and impurities in our pharma department. He graduated in chemical and pharmaceutical technologies, and he has been working in Neutron since 2016, gaining experience in the GMP regime. From 2023, is part of the extractable eligible unit in Neutron. Finally, the floor will be for Manuela Pavan, our external partner for toxicological assessment. She is a principal consultant in Innovatune, a science and technology-based company that provides consultancy services in silico toxicology in different contexts, including pharmaceuticals, agrochemicals, medical devices, cosmetics, and industrial chemicals. This is the agenda for today's session. So today's webinar will guide you through different main points on extractable eligible topic. We'll begin with the first introduction of the core principle of extractables and leachables, so what they are, where they come from, and why they matter. Then we'll move into the regulatory landscape, including key global standards and what's beyond the product compliance. So we'll explore how strategically design extractable and leachable studies. In the second part of this webinar, my colleague Mattia will greatly explain how to plan an efficient testing project. So we'll also take a look at the analytical methodologies used in the extractable eligible testing. From targeted screening to unknown compounds identification, you get a clearer pictures of how advanced analytical tools are applied throughout the process. Finally, Manuela will focus on toxicological risk assessment. So how do we determine if what we found in our testing poses a safety concern? You learn how both experimental data and predictive tools help fill knowledge gaps and guide safety decisions. Lastly, we'll wrap up today's session with some Q&A segment, so feel free to submit your questions in the chat at any time. With that, thank you for being here, and let's dive into what really lies beneath. But before starting, some words about Cotecna. To give you a brief introduction about our company, Cotecna is a Swiss-based company headquartered in Geneva, with offices and labs across the globe. It's a leading provider of testing, inspection, and certification services. Today, we now have over 7,400 employees with more than 180 offices and laboratories. Our trusted network of professionals and certified laboratories provides expertise across our 5 key sectors, namely agriculture, food safety, metals and minerals, consumer products, and system certification and ESG solutions. So some words about Neutron. Neutron is the biggest laboratory of the Cotecna Group, located in Modena in the north of Italy, with more than 550 employees working in 10,000 square meters of laboratory, but managing more than 1,000 samples per day and more than 5,000 accredited tests. These are the main fields of our testing activities, which Neutron has activated during its 50 years of history in relation to the market and in relation to customer requests. But going down to the real core of this webinar. So for sure, we must start with the fundamentals. So what are the extractables and leachables? Extractables are chemical compounds that can be drawn out of material when it is exposed to aggressive conditions, for example, high heat, solvents, or extreme pH. These conditions don't necessarily reflect normal use, but they are designed to stress the material and reveal what could potentially come out. Leachables, on the other hand, are the compounds that actually migrate into the drug product or into a patient under normal real-world conditions of use. These are the compounds that regulators are especially concerned about because they pose a direct exposure risk. Using the proposed definitions, extractable and leachables will be highly correlated with extractable representing worst-case leachables by both definition and intent. But the relationship between extractable and leachable is actually situational. It can vary from a full and complete correlation to a partial correlation, due, for example, to an interaction between drug product and packaging materials. Now, where do these compounds come from? They can originate from, for example, materials directly in contact or not with the drug or the patient. So think about containers, closures, tubing, filters, and so on, and even things like stoppers or labels. Common sources include plasticizers, stabilizers, additive, colorant, and so on. So many of these additives are necessary for performance or manufacturability, but they can become unintentional contaminants if not properly evaluated. So understanding the difference between extractables and leachables and identifying their potential sources is the first step in designing an effective extractable and leachable strategy. And as just explained, extractable and leachables can originate from several sources across the product life cycle, but which are the main actors? First, we have packaging materials, of course, like primary and secondary packaging, for example, containing closure system, labels, and cartons. Secondly, drug delivery systems such as inhalers, syringe have to be also taken into account. And these components in particular pose a higher risk because of their direct contact with the drug or the patient's body. Third, manufacturing and processing equipment like tubing, pumps, filters, and bioreactors can also be sources, especially in biological drug production, where contact surfaces are quite extensive. Finally, medical devices such as implants and surgical tools must also be considered. Therefore, understanding and identifying extractable leachables is an essential key point to protect both product quality and patient safety. First, extractable leachable compounds can directly impact the quality of a drug or device. They may interact with the formulation, alter its potency, or compromise the performance of a medical device. Secondly, and most critically, leachables can pose serious health risk to patients. Some of these compounds are toxic, carcinogenic, or can trigger allergic reaction even at very low concentration. Third, extractable and leachable can affect the shelf life and stability of the product. So unintended chemical interaction may cause degradation over time, leading to a reduction of the efficacy or even harmful bioproducts. Finally, there are real-world consequences. There have been notable product recalls due to extractable leachables. These events not only undermine patient safety, but also result in significant regulatory and financial repercussion. So this is why early extractable leachable assessments are not optional, they are quite essential. Regulatory bodies, including FDA or EMA, have established comprehensive guidelines and standards for the evaluation and control of extractables and leachables. Here you can find some of the key standards and guidelines, starting, for example, from ISO 10993, particularly Part 18, that focuses on the chemical characterization of medical device materials. It's a foundational requirement for example, for demonstrating biocompatibility. Then we have USP 163, which outlines how to conduct a risk-based assessment of extractables using simulation studies under exaggerated conditions. USP 164 complements that with guidance on leachables testing, focusing on what compounds may actually migrate under real-world use. And for combination product, USP 165 integrates this concept with device-specific considerations, providing an harmonized approach. And in addition, for USP 61.1 and point 2 focus on the characterization of plastic materials and packaging system respectively. These are vital for qualifying components used in the pharmaceutical containers, closures, and delivery system too. On the industry side, we see a valuable contribution from collaboratory groups like the PQRI, so the Product Quality Research Institute, that has published extensive guidance, particularly for orally inhaled and nasal drug products, where patient exposure risk is high. Similarly, the Bioforum Operations Group, or BIPOG, provides best practices for evaluating single-use systems in biologics. Their risk-based models are often referenced in regulatory submission. And looking ahead, the attention is on the next ICH Q3E that will offer global harmonization for extractable eligible evaluation in pharmaceuticals. All these standards collectively shape how industry approaches extractable eligibles, not just for compliance, but to ensure patient safety, material control, and data transparency. When we look at industry-specific expectation, they vary by product type. For example, for medical devices, chemical characterization and biocompatibility are essential factors to be evaluated, while for pharmaceuticals, the focus is largely on container closure system. And in biopharma, single-use components like bags and filters demand close scrutiny due to high extractable potential, and the focus is mainly on leachables identification. But in general, to proactively manage compliance, we recommend three main key strategies. First, plan extractable eligible assessment early in product development. This helps with material selection and reduces late-stage surprises. Second, collaborate closely with analytical and toxicological experts, especially when interpreting complex data or qualifying unknown compounds. And third, maintain a risk-based approach to align with global guidance. So clearly documenting the testing strategy, compounds evaluation, and safety threshold gives regulators confidence in the submission. The principle of risk-based approach also opens the door to an important and practical clarification. A well-structured risk assessment can allocate resources appropriately, with highest risk products receiving the most comprehensive testing regimens, while lower risk products may require less extensive evaluation. So a risk assessment is a critical first step in designing an appropriate extractable eligible evaluation strategy. In fact, according to the USP 165, factors such as patient population vulnerability, route of administration, and duration of the exposure carry the highest weight in determining the required testing approach. Therefore, the key is to evaluate in the first step the intention use and patient contact. Secondly, assess the material type and supplier data. In fact, the material is well characterized, and there is the availability. When there is the availability also of the supplier extractable eligible data or prior product history, this can significantly reduce the need for fresh extractable testing. Process parameters matter as well. Low temperature, short contact time, or non-aggressive solvent further decrease the risk of chemical migration. And all of these factors can be compiled into a risk-based justification document, clearly showing why the extractable risk is low and how existing evidence supports that conclusion. So in these low-risk scenarios, a targeted leachable assessment or sometimes no-forfeit testing may be sufficient as long as the justification is scientifically defendable. So we can say that this approach doesn't just help reduce unnecessary testing, but it also straightens development timelines, aligns with regulatory expectation, and ensures patient safety through evidence-driven decision-making. But what about when these assessments reveal the presence or potential for critical compounds? We must know that risk assessment is not a checkbox exercise. It's a critical decision-making tool. When it reveals the presence of critical compounds, it becomes the bridge to deeper analytical work through extractable and leachable testing. But make no mistake, extractable and leachable testing is not a routine testing. It's a multi-phase, multidisciplinary process that demands deep scientific understanding, advanced analytical capabilities, and strategic regulatory insight. In this case, as Neutron, we come up with a specific workflow, here shown, divided in different steps, each one fundamental to reach the best outcome. And to get to the heart of this complex word, I leave the floor to Mattia for a more in-depth look at strategic testing approaches and an overview of the analytical technique required. Mattia, please, the floor is yours.
Thank you, Valeria, and good morning to everybody. So, in this first part of my presentation, I would like to introduce a short definition of extractable and leachable, and make a further comparison between these two testing methodologies. So, extractable can be identified as compound that potential migrate from the packaging to the drug substance or the drug product in worst condition, so in aggressive condition. According to this, and focusing on the analytical point of view, the study of extractable was conducted in aggressive conditions in terms of temperature, in terms of pH, and also times of contact. In this study, we used the solvent called simulant for the contact with the package materials, which stress the materials and so extract all the component that can migrate potentially from the packaging. But in the same time, this type of solvent must be simulate the characteristic of the drug substances in order to have a correct comparison or a correct extraction according to the characteristic of the drug. The aim of the studies is the identification. So we got the scope is identify the molecules that were above a specific limit. And the analytical approach is an on-target approach with a screening analysis. On the other hand, leachable can be defined as a compound that real migrate from the packaging to the drug product. For this reason, the leachable studies is conducted directly on the drug product and in condition of in-use. So in the same condition that the drug product is used or storage. In this way, we can evaluate all the compounds that came out from the package, so real migrate compounds. And the aim of this study is the quantification. This because the study of leachable starting from the data obtained from extractable, and the focus are the molecules that were above the limit found in the extractable. The analytical approach in this case is a target approach, so specific molecules were searched. And anyway, it's also possible to perform a leachable study as an on-target study. in order to verify the absence of compounds in the drug product that the extractable study can't to detect. So at the end, I would like to underline the importance of these two methodologies of analysis. And in my opinion, these two methodologies are not parallel, but we must manage these as the same, as a part of the same landscape. And so it's very important the cross-reference of the data obtained from extractable and leachable, because very often a result obtained in the leachable can be clarified, or we can give the source of this compound looking at the extractable data. So, speaking about the analytical part, extractable and leachable study can be defined as a multi-determinative analysis that involves a various technique in order to detect different families of compounds, such as volatile organic compounds, non-volatile organic compounds, and semi-volatile organic compounds, detected with different technique, for example, LC-cutoff for non-volatile, GCMS for volatile and semi-volatile organic compounds. This family were detected with an on-target approach, but we have also a family of compounds detected with a target analysis. For example, elemental impurities detected by ICP-MS and anions, for example, detected by ion chromatography. From the analytical point of view and evaluating data, the key point is the IT, also called analytical evaluation threshold. This value is calculated taking into account the characteristic of the drug, so the number of doses per day, the volume of doses, and so on, and represent the edge of the evaluation of the data. So, as you can see in the image, for the molecules below this limit, is not necessary an identification or a safety evaluation. But for the molecules above this limit, is mandatory the identification or a safety assessment. In conclusion, apply always the lowest possible limit is not necessarily the best choice, but it's important to use the real parameter in the calculation of IIT in order to give a correct evaluation of the data. So, waiting for the harmonization of ECH Q3A, we implementing in our analytical offer also the evaluation and the analysis of common nitrosamines detected by LC triple quadruple. And we performed the analysis of 15 nitrosamines following the EMA list and applying the limits detailed in this list. This type of analysis can be help us to have a wider view on the compounds that can migrate from the packaging. So, in this second part of the presentation, I would like to share the Neotron proposals for elemental impurities analysis that can be divided into different approaches. The first one is the single product testing. Here, the study protocol is focused on a single drug product, evaluating all the information from the package leaflet. So the data evaluation is referred to the IT value calculated on this specific product. And the result obtained represent a sort of fingerprint related to the specific studies. This type of study is very interesting for pharmaceutical company because help to evaluate the impact of all the steps of the production process of the drug. On the other hand, The second approach is called a smart testing strategy. Differently from the first one, the study is optimized to cover a wide range of product without specific information of dosage or route of administration. But the focus is the same contact material or the same packaging. So in this case, we don't calculate or use the IT, but the data evaluation is referred to a value that we call it SQAT, or semi-quantitative evaluation threshold. And in this case, we exploit all the maximum instrumental sensibility, and we perform a lower limit as possible in order to obtain an adaptable profile. So the results are freely adaptable to different drug product, different dosage related to the same containing closure system. So this type of analysis is relevant or important for packaging supplier companies. So at the end, I would like to show a typical roadmap involved in a target leachable studies. This type of activity can be divided in 3 different steps. The first one and most important and critical step is called feasibility setup. And in this phase, we set and we verify the parameter that we obtained from the extractable data in order to apply this on the drug substance, and with the scope to eliminate all the interferences derived from the matrix. The second step is the method validation. So we perform a full validation according to the ECH parameter, following a validation protocol, and at the end we obtain a routine method of analysis. This method will be applied in the last part of the studies during the shelf life of the product. This shelf life can be an accelerated study or a long-term stability study. And the aim of this part of the analysis is verify the absence of changes in the leachable profile. So so at the end of all analytical activity, if some unleachable compound were identified and quantified above the IT, the question becomes, my product is safe or not? For answering to this question is essential the collaboration between analytical scientists and toxicologists in order to have a complete and wider evaluation of the data. So teamwork is necessary to bridge the gaps of the 2 roles and take the correct decision and the correct evaluation of the data. For this reason, we collaborate with a team of experts, such as Manuela Pavan, Principal Consultant of Innovatune, who will show in detail the importance and the construction of a toxicological assessment. Thank you for your attention.
Thank you, Mattia. Now, let's move for the final topic with our third speaker, Manuela Pavan. Please, Manuela, the floor is yours.
Thank you, Valeria. My presentation today focuses on the toxicological evaluation of extractable and leachables. And as Valeria already mentioned in the previous talk, assessing and controlling the toxicological risk of extractable and leachables is essential to make sure that the pharmaceutical products are safe, effective, and high quality. However, despite the critical role of extractable and leachable assessments in pharmaceutical development and life cycle management, the current regulatory landscape lacks a comprehensive and globally harmonized framework that specifically addresses the identification, toxicological qualification, and risk-based control of these substances. There are existing ICH guidelines on impurities, for example, the ICH-M7, the Q3A, B, C, and D, that they all offer relevant principles, but were not specifically designed to cover extractable and leachables. Next. In the absence of a dedicated regulatory framework, best practices have been proposed by organizations like the Product Quality Research Institute and the LC Consortium. For example, the PQRI proposed a workflow for parenteral drugs that sets safety limits based on mutagenicity and sensitization potential. Mutagenic leachables are limited to 1.5 microgram per day, while non-mutagenic substances are further assessed for sensitization, where potent sensitizer may then require limits below 5 microgram per day, while non-sensitizer may also allow higher threshold based on available toxicological data. And the LC Consortium also contributed significantly in the extractable and leachables field by promoting best practices and providing input to support risk assessment of extractable and leachables. Next. To address the currently regulatory gaps, the International Council for Harmonization is developing a guideline that was already mentioned, the Q3E, which is going to focus on the assessment, testing and control of extractable and leachables, and which is expected hopefully in June 2025. According to the 2020 concept paper, the Q3E will provide a globally harmonized framework for extractable and leachable evaluation. It will complement existing ICH guidelines. It will provide the science and risk-based thresholds for the identification, qualification, and reporting of extractable and leachables, and it will also clarify the use of existing data and prior knowledge to determine the real need to further testing. And overall, the final aim of this guideline is expected to improve consistency among regulators and industry. Next. In this slide, I summarized the current workflow that is used for toxicological evaluation of extractable and leachables based on current best practices and pending the release of a formal ICH Q3 guideline. So it's something that may be reviewed based on the new guideline. The assessment typically begins by searching whether a regulatory limit has already been established for a given compound. If that is the case, the limit is reviewed to determine whether it is appropriate for the specific use case, considering factors such as the regulatory context in which it was originally proposed, but also route administration, duration of exposure. If no regulatory limit is available, the evaluation then starts with assessment of genotoxicity. This is always the key endpoint that is assessed, following the principles outlined in the ICH-M7 guideline. And if the compound is found to be genotoxic, either experimentally or computationally, then the default limit is set to 1.5 microgram per day, in line with M7 guideline. However, compound-specific limit may also be derived. For example, in the case carcinogenicity data are available for that substance, an acceptable intake may be established based on those specific data. If the substance is not mutagenic, a weight of evidence approach is generally applied to evaluate its potential for irritation and sensitization. And if it is identified as a potent, strong sensitizer, a conservative threshold below the 5 microgram per day is generally applied. While if the substance is not sensitizer, the default limit of 5 microgram per day may still be used. However, this limit can also be increased if compound-specific toxicological data are available. For that purpose, the next step consists in a comprehensive literature review that is conducted in order to characterize the compound toxicological profile. And this includes reviewing data on key endpoints, including mutagenicity, repeated dose, carcinogenicity, reproductive and developmental toxicity. Then, based on the quality and quantity of available data, the compound can be classified as either data-rich or data-poor. In both cases, a compound-specific safety limit, like, for example, a permitted daily exposure, can be derived. But for the data-rich substances, this is done directly from the toxicological data of the compound itself. While in the case of data-poor substances, the limit is derived based on data from a suitable analog, meaning a similar compound, using the read-across approach. And I will get back on this in the next slide. So in the following slides, I want now just to go a little bit in more details on each step of the workflow. Next. As we mentioned, if the regulatory limit is not available, the first key step consists in the genotoxicity assessment according to the ICH-M7 guideline for drug impurities. The goal of this guideline, or of the hazard assessment of the guideline, is to classify each impurity based on its potential, based on its mutagenic potential. So each class corresponds to an appropriate control strategy, defining how much of that impurity is allowed in the drug. A key concept in the guideline is the TTC, or the threshold of toxicological concern, which is set to 1.5 microgram per day for mutagenic impurities, assuming a lifetime exposure. So the M7 workflow begins by evaluating the mutagenic potential impurity by searching data, experimental data, focused on bacterial mutagenicity and carcinogenicity. And if high-quality data are identified, the target impurity can be classified as class 1, mutagenic and carcinogenic, class 2, mutagenic with a known carcinogenicity potential, or class 5, if it is known to be not mutagenic. However, if experimental data are not available or not sufficient, a QSAR assessment and in silico approach can be performed, and based on that, if a positive outcome is given, the impurity is assigned to class 3, which is mutagenic. If a negative outcome is given, then it can be classified as class 5, not mutagenic, or class 4 in the case of a given alert that is shared with a known negative, experimentally negative compound like API or related impurity. Next. Thus, the M7 guideline supports and strongly also promotes the use of computational approaches. So these are known as in silico methods, QSAR approaches, the quantitative structure activity relationships, but all these terms refer to computer-based models that are used to make predictions, to predict the toxicological property starting from the information of the chemical structure of a given target compound. In the specific case of a guideline, the use of a complementary, the guideline suggests the use of two complementary QSAR models. One is an expert rule-based system, the other one is a statistical approach, and both the two models need to fulfill the 5 OECD principles for QSAR validation that were defined to set the criteria that ensure predictions are scientifically sound and acceptable for regulatory purposes. And the use of the two different approaches is designed in order to increase the reliability and support a more robust classification in the cases where experimental evidence is not available. Next. A number of computational tools have been developed to support this type of assessment, incorporating the two different approaches. I do not want to go here into the details, but just to share with you some general considerations. So these tools, the models, need to be built on a broad and well-curated training set, ideally combining both public and proprietary data. These data sets also needed to be regularly updated in order to reflect new findings that may be available, and they also needed to be based on a robust algorithm that provide accurate and reproducible predictions. Transparency is also another key element that is needed for a tool to be used for regulatory purposes, and all the tools need to be recognized and accepted by regulatory authorities. And here you find just mentioned some of these of the most commonly used tools for M7 assessment, grouped depending on the specific approach that they cover. Next. According to the M7 guideline, an expert review of both experimental data and in silico predictions is always strongly recommended in order to strengthen the confidence of the final outcome. In fact, the expert review can support the relevance and or the reliability of a positive or negative prediction, can provide a rationale to conclude on equivocal results. So when results are not so clear from the outcome of the computers or the models, and can also be used to overturn a prediction. The expert review typically involves a number of key elements, including the analysis of the quality of underlying data, the identification of reactive or mitigating structure features, the evaluation of information coming from analogs, and the assessment of the overall consensus among and between the two models applied. Next. Just as a side note, just to keep in mind that in the context of M7 genotoxicity assessment, we need to remember that there are certain chemical classes like the nitroso compounds, the alkyl toxic compounds, or the apelcotoxins that are considered a part of a cohort of concern. So for these substances, the general TTC of 1.5 microgram per day does not apply, and the more conservative limit must be applied. Next. According to the workflow, in the next step, if the target compound is assessed as not mutagenic, the next step consists in assessing its sensitization or irritation potential. This can be performed, of course, based on experimental data, if available, and if an extractable and leachable is identified as a potent or strong sensitizer, according to the PQRI workflow that I previously mentioned, the threshold is generally set below the 5 microgram per day. In the absence of experimental data, a weight of evidence approach can be applied where in silico models are generally used together with an analog approach in order to draw conclusions regarding the irritation and sensitization potential of the substance. For sensitization, it's also important to note that the current in silico tools are available to predict the skin sensitization, typically based on LLNA data, which, however, is considered as a reasonable indicator for systemic sensitization, as it is discussed in the literature. If the substance, next please, is not mutagenic, but is also not a potent or strong sensitizer, then we can move forward and we can derive a compound-specific limit based on the specific toxicological data of the substance. And to do that, the next step consists in conducted a comprehensive literature review where multiple sources are consulted in order to gather toxicological information on the target compound. And the focus here is on key endpoints, specifically and often repeated dose toxicity, carcinogenicity, rape or developmental tox studies. And based on the outcome of this analysis of this review, the compound can be classified as a data rich. In that case, it means that we have sufficient toxicological data available for the target substance itself, or data poor in the case the information is lacking or is limited. Next. When a compound is classified as a data rich, it means that we have access to sufficient high-quality toxicological data that allows us to derive a compound-specific limit. The typical example is the permitted daily exposure, the PDA, and this value is calculated directly from the experimental toxicological data that are available for that specific substance. However, in some cases, rather than working and deriving a limit from an individual compound, substances can be grouped based on the fact that they share some similarities in the chemical structure or in the toxicological behavior. And for these groups, a class-specific limit may be defined. So this approach is specifically useful when the data are limited for an individual member, but robust information exists for the class as a whole. And importantly, in such a case, computational methods can support the class limit derivation in a number of ways. For example, identifying structural alerts, assessing biological similarities, identifying patterns across the clusters. And I would also like to say that this is an emerging field where scientists are actively conducting research. Next. As an example of this type of research, we have been collaborating with industries like Pfizer, Johnson & Johnson, and FDA on an analysis of a data set comprising approximately 1,200 extractable and leachables. The analysis was presented as a poster at the SOT 2025, and I will be happy to share the poster with you in case of interest of that. The objective of this work was to explore how these compounds are distributed within the conventional extractable and leachable chemical space and to cluster them based on structural characteristics. And for example, this analysis highlighted that, and one of the key findings was that approximately 50% of the compounds are data poor, meaning that they lack sufficient toxicological information to derive a direct risk assessment. However, the clustering process can help, for example, in identifying suitable analogs to support a rigorous approach for these data poor substances. And I will focus on this in the next few slides. Next. So the read-across approach is one of the most widely used methods to fill in data gaps in human risk assessment. And in the read-across approach, the information on a given property for one or more sources, meaning data-rich chemicals, is used to derive the same information, the information on the same endpoint for one or more target chemicals that are lacking this information, so the data poor chemicals. The underlying hypothesis of this approach is that the target chemical is very similar to the source chemicals, based on a number of features like structure, physicochemical, metabolic, and the toxicological properties. And then if that is the case, so if the similarity is sufficient, then the property of the effects of the target can be inferred from the source chemicals data. Next. A critical step in this process is, of course, the initial identification of suitable analogs. The analogs need to meet two criteria. They need to be similar to the target compound, as we said, according to different line of evidence, but they also need to have robust toxicological data that are adequate for the derivation of a compound-specific limit. Next. In this slide, I'm illustrating a case studies that focuses on the isobutyl dipate. This is a compound with limited toxicological data included in the PQRI database. There were experimental data showing that the compound is negative for bacterial mutagenicity. Also available information suggests that there is no concern for skin and eye irritation and skin sensitization. However, due to insufficient data to directly derive a compound-specific limit, a quantitative limit was established by using a read-across approach. Next. A suitable analog was identified. The diethyl adipate was selected as a suitable analog based on its close structure similarity to the target, but also based on available robust toxicological data, particularly for repeated dose and developmental toxicity studies. There are NOEL data that were identified from multiple regulatory sources and used to support the read-across justification and to derive an acceptable exposure limit for the target compound. So this example is just to give you an idea of how, in the case of data poor substances, a safety limit for the target compound can be inferred from the toxicological data of a suitable analog using a scientifically justified read-across approach. So to conclude my presentation, I would like to say here that as it was commented in this talk, but also in the previous one, the toxicological assessment of extractable and leachables is and remains a critical step in order to ensure the safety and quality of pharmaceutical products. And although there are existing ICH guidelines that provide valuable frameworks and relevant principles to handle this topic, they do not fully address the extractable and leachables. And the ICH Q3E guideline is expected to fill in this data gap by introducing an harmonized and science-based strategy for extractable and leachables. And where experimental data are limited, I showed you how computational toxicology, read-across, in silico methods, always supported by expert interpretation and expert review, can be used to offer a pragmatic and defensible path forward. Finally, I would like to acknowledge and to thank all my colleagues who were involved in this work, and of course, all of you for your attention.
So thank you again, Mattia, and thank you, Manuela, for your very interesting presentations. Now it's time for Q&A sessions. So we have received some questions during the registration process, and we have selected a few of them that we will now answer with you. But if during the presentation you collected any curiosity or need further information, please feel free to use the Q&A section or chat box. So starting with some questions. We have seen that a limit of 1.5 microgram per day applies to genotoxic impurities, whereas a limit of 5 microgram per day to sensitizers or irritant compounds. Could you explain the scientific rationale behind these specific limits and how they were derived? Manuela, this is for you.
Thank you, Valeria. Yes, the default limits of 1.5 microgram per day and 5 microgram per day associated to genotoxic and sensitizer compounds, respectively, derive from the adoption of two different approaches. The limit of 1.5 microgram per day is based on a TTC approach, a TTC concept, a threshold toxicological concern, which is a risk-based approach that is used to estimate the daily intake of mutagenic compound associated with only negligible lifetime cancer risk. On the other hand, the limit of 5 microgram per day is derived based on a statistical review, a statistical analysis of toxicity data, representing the level that is generally considered safe and protective for the majority of patients. So these two thresholds, these two different limits, are derived from two different approaches. One, as I said, is a calculated lifetime cancer risk, while the other one is derived empirically from skin sensitization, and irritation data.
Thank you, Manuela. So another question. Okay. You mentioned that the identification of suitable analogs is a critical step within a Red Cross workflow for extractable and leachables. In practice, how is this search performed? Manuela, this is for you again.
Thank you. Yes, indeed. I just mentioned quickly during the presentation that the retrieval of similar compounds with adequate data to use as analogs for the Red Cross is really crucial in order to properly apply this approach. A pragmatic approach that is often applied is first to search within comprehensive genotoxicity databases, focusing therefore on compounds that have AIMS data. The reason of that is that indeed the mutagenesis is one of the most frequently evaluated endpoints among the data-rich compounds. So if we start from there, we are more likely to find compounds with a good amount of data available. Consequently, the search could be extended to other relevant databases to gather, for example, additional toxicological data that are needed to derive, for example, a PDE limit.
Okay, thank you again, Manuela. So, another question. Okay. Which are the key steps in extractable and leachable risk assessment? I can give you an answer for this question. So the extractable leachable risk assessment starts by material identification for sure. So evaluating the list of all materials in contact with the drug product, for example. Then we must move to the extractable study, identifying and quantifying all potential extractable found in a supplier study and drop into the product that is under focus. If any concern compound is revealed during this simulation study, a toxicological risk assessment is needed to evaluate safety, and eventually mitigation and control must be implemented if any risk exceed acceptable levels. So another one. Okay. In which situation can be useful to perform an untargeted leachable study? Mattia, I think this is for you.
Yes. In my opinion, the untargeted leachable study can be helpful for different application. For example, I think about little changes in the container closure system of a drug product. So, for example, changing the adhesive, the composition of the adhesive of a label. In this case, the untargeted leachable study can help to verify that these little changes not impact on the drug product. So on another example, the change can be the changes of the secondary packaging. All these type of minor changing can be investigated, in my opinion, under an untargeted leachable studies, and represent a sort of addendum of a leachable extractable study performed before.
Okay, thank you, Mattia. There's another question, I think. Okay. how can it be possible to manage an extractable study when it's necessary to reach a very low AET? Mattia, is again for you.
Thank you. Interesting question, because it's often in laboratory we faced with this problem, with this situation. So we can solve this situation maybe applying a sample preparation strategies. For example, concentrate the medium of extraction, or for example, maximize the surface of contact of the packaging with the medium. So in this case, with these solutions, we can perform analysis with low AAT.
Okay, thank you, Mattia. Now we have some questions in the chat box. So, what is the adoptability of BPOG guidelines in extractable leachable evaluations? I can answer to this. The BPOG is representing guidelines for single-use system. For example, for tubings, stoppers, or pumps, or other parts that must be interesting for the equipment in the process of the drug production. And these guidelines gives some idea of which can be the good solvents, so the good simulants, as said before by Mattia, or which can be some other useful tool, for example, the good temperature and the good timing duration of contact with this simulant, in order to have the best outcome for each of any kind of contact materials. Then another question is, during the presentation, you always refer to drug products. Are extractable and leachable studies required for drug substances as well? Mattia?
I think that obviously the analysis of extractable and leachable can be performed and can be referred to the drug product, for example, drug substances. For example, an API stored in a bag can be studied from the extractable and leachable point of view. So in my opinion, yes, it's important, this type of study, also in drug substances.
Thank you, Mattia. Okay, we are now at the end. So we hope that you find this webinar helpful in clearing your doubts and curiosity on this complex topic. And I want to give a big thank to our speaker, Manuela and Mattia, for sharing their great expertise. And thank you to Dana Sanchez and all Cotecna team that help us in the organization of this event. And of course, thank you all for joining us today. Some information, we will send out an email in the coming days containing a survey form, a recording of this webinar, and the relative presentation. Here you can find Cotecna and Neotron contacts, so feel free to contact us for other questions and requests. So have a great day, everyone, and see you to our next webinar. Thank you.
Thank you to all.
Thank you.
Bye-bye. Have a nice day.